Hello everybody, welcome on the last beach vlog this year, 2023, and I hope you really enjoy still great weather in Netherlands if you're based in Netherlands, and all those which are not in Netherlands, I hope that you really enjoy this vlog, this last, I think, beach vlog this year, and we're going to talk today, and I'd like to actually bring more attention about stem cell based therapies. So this is this delicious game changer in regenerative medicine, and which I brought during last vlog. And I'm really glad that today is not so windy, otherwise I would have to really shout a lot to compete with the wind, which generates actually a lot of noise. So I'm really happy that we have beautiful, as you can see, not windy weather. And regarding stem cell therapies, I thought this is very important to actually talk about also clinical trials, because I told you last time about stem cell tourism, that there are people which are desperate, which look for experimental stem cell based therapies in order to be treated. And I think this can have very bad consequences if you enter wrong clinic. And that's why I thought it's good to actually zoom in a little bit more on stem cell therapies and clinical trials and what has been done so far, and what type of diseases has been treated. So I've been very much inspired by the article of scientists and actually medical doctors from Stanford Medical University, and I will give you a link in the comments. But this article was bringing a lot of interesting data, and that's why I have to sometimes look back on my laptop here to bring the numbers and don't mess it with the numbers. So first of all, what is important about stem cells, that you can compare it to interventional oncology, where dose of basically the drug is given systemically, so intravenously, and this drug is distributed through whole the body. Therefore, you can imagine if the dose is very high, this drug is actually very cytotoxic, talking plain language, very toxic, not only targeting cancer cells, but also targeting healthy cells in our body through our circulation system. And that's what you don't want. You want a therapy, and that's why you could compare it to, in a way, chemotherapy. But in terms of stem cells, you want to have therapy which is not just delivering stem cells through whole blood system everywhere, but you want to actually have stem cells which are injected in the organ which is requiring repair, and therefore stem cells can regenerate this organ. So that's why this article brought great introduction of interventional regenerative medicine. So the one which is with minimally invasive procedures delivers stem cells into targeted organ which is damaged, which requires a regeneration or repair. And I think that's very important to realize that. So if we think about terms, I think it's important what I would like that you remember from this vlog is that there is interventional regenerative medicine, and I think that's our future in regenerative medicine, right? To deliver stem cells to target organ which requires repair or is damaged. And of course, you probably think of, oh yes, Aneta is stem cell biologist, she knows a lot about stem cells. Yes, I know a lot about stem cells. I'm fundamental stem cell scientist, preclinical. And you have to remember that whenever you think about therapy, you have to think not about fundamental research or preclinical research, which mainly is done on animals or animal models, but you have to actually if you want to introduce therapy successfully, you have to think about clinical trials, and there are different phases of clinical trials. And maybe another vlog I would talk about per se what type of clinical trials are there and what are the stages. But today I would talk in general about clinical trials, and what was important to realize, and I think here stress about, that in contrast to preclinical research which is done on animals and to develop effective stem cell based therapies, there is a lot of success in the studies, but there is a lot of also discrepancies compared to clinical trials which are done on patients, on human patients. And I think that's the first important lesson, just to make it short, because there is a lot of examples maybe of very well aligned studies and results, but many of these preclinical data are not relevant to clinical therapies. So it's actually not something which you want, because you do preclinical studies to take these results with you when you want to develop clinical trial and clinical treatment and apply it to human patients. Still, this is something which is not helping. Preclinical data are very different than clinical trials. And I will explain later why it is that. But you also want to develop successful stem cell-based therapy, which in the end brings effective treatment to human patients. So brings effective treatment and develops successful clinical trials. But before we go to this, because I would like to give you some examples, of course we have to realize what type of potential diseases stem cells can be used to develop successful therapies and treatment. And here I'd like to stop for the moment on blood stem cells. So many times you can find the term hematopoietic stem cells, HSCs, and blood stem cells has been developed, or has been actually not developed, but has been characterized the best from all different stem cells which you can find in our body, in our organs, in our tissues. And they were also, because they are in our circulation system, they can be found in our circulation system, they can be also easily injected intravenously, so systemically, to treat hematopoietic system disorders or hematopoietic hematological disorders. And so far I'd like to only mention about hematic stem cells that so far there have been currently 14 approved cellular products, including blood stem cells, which involve allogenic umbilical cord blood derived hematopoietic stem cells or blood stem cells, and one which use autologous chondrocytes for knee cartilage defects. And of course you may already think about what does it mean, autologous, and what does it mean allogenic. Autologous comes from actually the patient itself. These are your own stem cells, and if you heard maybe about cord blood banks, when you're born, your umbilical cord blood is actually processed, and stem cells from there, blood stem cells, can be stored for the child for later, and if needed. So that's what autologous source of stem cells is. And allogenic are the ones which come from other donor of stem cells, and but they are still well matched. So that's a little bit about summary, because I'd like to also not only talk about preclinical research, but also what is the current update on clinical trials on blood stem cells and products which are in form of stem cells delivered and used in this clinical treatment and clinical trials. So that's something about blood stem cells. And another source of stem cells, which you might heard, and comes from actually liposuction, I can tell, are mesenchymal stem cells. They can differentiate into bone, fat tissue, and cartilage. And they have been shown to be very successful source of stem cells to use to treat actually different type of diseases. And that's what I like to actually stress about this stem cells here. And I will now move to diseases, because I told you that I would like to mention important diseases which stem cell therapy can be used for. And one of these diseases is cardiac disease. And as maybe you have heard, cardiac actually disease is leading cause of death globally. So that's very important disease to find a treatment, successful treatment. And stem cells are offering great actually potential to treat cardiac diseases. And what I'd like to tell you a little bit about cardiac diseases here is that, as I told you, you can have stem cells which come from the same tissue, so from the heart. You can have cardiac stem cells, but actually MSCs have been very successfully used in preclinical studies. Another type of option of stem cells are the ones which are genetically reprogrammed or genetically manipulated. These are induced pluripotent stem cells, so IPS, which you can actually differentiate into heart cells, and we call it cardiomyocytes. And what happened if we think about cardiac disease? So MSCs have been used, and they appear to be quite, as I told you, giving good therapeutic effect, but it really depended the way they were injected. So the route of administration, so type of injection, how they were given to. And also cardiac stem cells, so heart stem cells have been used with some good results. However, this stage is still at very early, if you think about clinical trials, and successful clinical trials is still at very too early to really make strong conclusions about that. One thing is that a lot of studies have been addressed at preclinical level, but not at the clinical level. And especially if we think about IPS, and I think I have to take some drink. IPS, one of the things is that they are genetically manipulated, and they have been shown actually to carry some risk when they are injected of tumor genesis and cardiac arrhythmias. So that's why whenever you think of, are IPS able to be successful source of stem cells for therapy of cardiac disease, it's still under big question mark, because you have to first make sure that these cells, this IPS, are safe before you introduce it to human patients. And I told you about these types of administration, that depending how you inject stem cells, there can be different effect of the stem cells in therapy. And appear to be that in terms of cardiac disease and stem cells which have been used, route of administration, so more targeted intramuscular, appear to be better than systemic one, so which is done intravenously. So that's important thing important result. And also, if you think about stem cells, well, once we inject them, we want that they stay in this damaged organ, because they have to still differentiate to repair damaged tissue and replace the cells, tissue-specific cells or organ-specific cells. So they need to stay long enough. So this is called retention time, has to be long enough. And that was, oh, I have helicopter. So let's pass this moment for the moment. But this means that, yes, depending on the type of injection, retention time of stem cells can be very different. And compared to the systemic injection, actually MSCs in this case appear to stay 3 to 5 fold. They had greater retention after delivery. So that is really great result. And also the size of infarcts of the damage was decreased. So this is definitely very good result in terms of type of administration, right? Intramuscular, and we need to actually think about concentrating on this type of injections. And to summarize, we have currently 45 of 51 active and recruiting clinical trials, which have specified of targeted approach to stem cell delivery for the treatment of cardiac disease. Another type of disease, am I going with time? I think I will have to summarize it shortly. Stem cells have been also used for treating liver disease, especially MSCs show great promise in clinical trials, even in acute liver failure. And to summarize, we have 4 out of 18 active and recruiting clinical trials for specified, so targeted approach of stem cell delivery for the treatment of the liver. There is also stem cells have been used for treatment of diabetes type 2, and what I'd like to now more focus would be treatment also of stroke by not only MSCs and umbilical cord blood MSCs or derived from bone marrow, but also neurostem cells. So what very much got me interested that neurostem cells, so these ones which actually are derived or are present in nervous system, in the brain, can support the best regeneration through neurogenesis via direct integration of or stimulating endogenous proliferation, so amplification of cells, also stimulating, promoting angiogenesis, so vascularization, and axonal growth, so growth of neural cells. And these cells have been so far shown the most successful. However, if we look at animal studies, we again look at different types of injections. One of the injections which really got me interested is intranasal injection, so via nose, where stem cells can actually cross blood-brain barrier, which we have in our brain. So it's sort of filter system which doesn't allow compounds to cross the brain. It's very selective. And that is very interesting in terms of developing stem cell-based therapy via intranasal, in a way, delivery of stem cells, on neurostem cells. And this shown to be, at least on preclinical studies, very promising, with very promising results. Especially delivery also of mesenchymal stem cells, which are bigger than neurostem cells, to reduce cell death in infarcted regions of brain in rats. That also was very important to know. So to summarize, I think the future of interventional rich medicine is very bright, especially in clinical care of patients, but by enabling or allowing targeted delivery of stem cell therapy, so not systemically to damaged organ, with using minimally invasive approaches. But we cannot forget, as also inventors, scientists, that to deliver this stem cells targeted, very often sometimes intramuscular, we need to also, or in the heart or in the brain, we need to also have specific specialized delivery systems like microcatheters or balloons which are mounted with microneedle. So that helps to, usually stem cells are injected with the needle, and you can imagine the needle is very narrow, so it can induce a lot of mechanical stress of stem cells, which is called shear stress. And if we are able to deliver via different delivery system which doesn't stress mechanically stem cells, that could be the ultimate goal here. So there's a lot of, I think, here space for innovation regarding these devices to deliver stem cells to targeted regions without damaging stem cells. And of course, if we think about interventional rich medicine, we would imagine that we can produce these stem cells, store them in clinical grade conditions. But we also have to think now that we need to also expand them, right? Sometimes we need a dose which is very high, and we have to expand stem cells outside of the body, so in vitro conditions. And we also have to have proper standardized procedures, because as I told you, for IPS there is no good standardized for clinical trials procedures to differentiate them into fully mature cardiomyocytes. So we have to have standardized procedures of differentiation, of differentiation. Regarding also interventional rich medicine, if we think about this, we also have to think about something which has been created, like Interventional Regenerative Medicine Hub in California Institute for Regenerative Medicine, and where you have integration of stem cell biology, but fundamental biologists. You have you have medical doctors, you have clinical, of course, experts, you have healthcare experts, so you have also hospital infrastructure. So to make sure that the stem cells can be expanded under standardized clinical grade, in a way following the standard clinical grade procedure, and delivered as soon as possible, because time here also matters. In terms of delivery. And of course we should have very specific, depending on disease, if it's cardiac disease, if it's a patient suffering stroke or kidney failure or liver, develop liver disease, we have to really think about specific devices supporting and actually delivering successfully the particular stem cells to the targeted, so damaged organ. And I think when I read this article and I imagined this whole infrastructure, this seems to be a very multidisciplinary hub which combines stem cell biology, so fundamental science, with clinicians, but also interventional surgeons which are able to perform such delivery. And that's also another type of actually profession in the surgery where we have interventional regenerative medicine surgeons able to deliver stem cells to different organs, damaged organs in our body. I think this is something which I would like today talk about. I'm only maybe could give you some example how, because I thought about discrepancy and I like to come back to it. Future is bright, but we have to still looking at clinical trials, also think about standardization of clinical trials, getting enough open access information so that we can compare clinical trials. But even comparing results of preclinical studies and clinical data or clinical trials data. What was striking for me that, for example, there was discrepancy in cell therapy in intra-arterial MSC-based treatment of acute myocardial infarction, so cardiac disease. And one trial compared dosages of, if I think about that, of 12.5 million or 25 million single injection, where other opted for 1 million only per kilogram. So the differences are 12-fold if you think about this, or 25-fold, and this brings a perception that, well, is this a way how we develop clinical trials? And I think here I like to mention one of the companies, which is in Leiden Bioscience Park, I think having the best expertise in clinical trials, Galapagos, because what you have to also realize, there are also at university medical centers research groups which want to work on stem cells, but they want to develop clinical trial. And I've been on a meeting where what happened, actually it was very interesting to see that this research, which was developed for basic study, it was well developed, but this was not well developed for clinical trials. And then you see scientists or group leaders which try to reach companies which are performing clinical trials, which are specialized in clinical trials, to get their expertise. And then what happens after is that appears that actually this whole research, preclinical research, has been completely designed completely wrong for the clinical trial. So it's actually not really something which we can use to build actually fundamental data, which we can use later for clinical trial to start with. So we have to redevelop whole basically experimental design from fundamental research, from preclinical research, again, because that was done by fundamental scientists, not by research or clinical researchers or clinicians. And that makes it very difficult then to combine this data, which were already generated on animals, and now bring it as a cooperation, as a collaboration with, for example, a company like Galapagos. So I'm actually warning people here, when we think about clinical trials, development of clinical trials, before you start your research with perspective to bring the to develop stem cell therapy at the university and bring it to clinical trial, please also have a very strong already collaboration all the way at the early stage, from the early beginning with Galapagos or companies which are highly specialized in clinical trials, because this will save you time, costs, animals, and this becomes way more effective by collaborating with such companies. So that's what I'd like to actually share today with you as the last piece. And I believe strongly in interventional regenerative medicine and stem cells, MSCs, especially for treatment of lethally actually diseases which cause which are a lethal cause globally. So cardiac disease, stroke. So that's something which I hope mesenchymal stem cells, neural stem cells, and umbilical cord blood stem cells can be applied for. And I hope that we will work as a scientist, but also as a clinicians, as a companies, to develop effective clinical trials, so we can develop effective treatment for every patient. So that's my wish, that's my golden wish. I hope this will come true. It requires a lot of multidisciplinary cooperation between universities and companies and hospitals or academic medical centers, and definitely I believe that this is possible. So especially in Leiden Bioscience Park, we have complex of LUMC and companies, Galapagos, startups, and we can all combine our efforts to develop, in the end, from basic research, successful clinical trials, and bring these effective therapies globally, because that's also important to bring them globally. So I hope that you now got more insight about stem cell based therapy, so the delicious game changers in regenerative medicine, and that you will get inspired, and especially for people which look desperately for stem cell treatment, please also watch this vlog. If you have any questions, please let me know, because it's important to bring to you from me as a stem cell biologist, scientist, the awareness about the current status and how regenerative medicine is evolving towards treatments of different important diseases globally. So I think if you have some questions, please put them in the box, in the comments. If you have some comments, if you have some articles which can be also interesting, I'm really happy to actually also take a look and also keep discussing this subject. This is very important subjects, because there is a lot of discrepancies, and preclinical data also show a lot of they're actually misaligned with clinical data, and that's something which we have to find a way how we can use preclinical studies and results to develop effective clinical trials for stem cell based therapies. So I think, yes, that would be the whole content. I know that maybe it was not easy to follow, but I try to bring it in plain language and explain it, so by giving examples. So I hope it's clear. If it's not, also please let me know. I'm looking forward to your questions. So from here, from this beautiful place, from Hague, from Scheveningen, I'm sending you very warm greetings, and see you next year on the next Agile Beach vlog. Bye!